Okay, let's get started. Hi, everyone. Thank you for joining our webinar today. My name is Chiming Wang. I'm the International Marketing Director at Cyagen. I'm the moderator for today's webinar. Joining me today is Lily Fan. Lily is our cell and gene editing platform manager here at Cyagen. Lily has over 9 years of expertise in iPSC reprogramming, gene editing, and differentiation strategies. The title of her presentation today is Application of iPSC Gene Editing Technologies in Biomedical Research, in which she will talk about some of the challenges and our solution to it. During Lily's presentation, you can type in your questions in the chat box. We will answer as many as we can at the end of the presentation. Without further ado, I will pass the stage to Lily.
Okay, thank you. Welcome to today's webinar on applications of iPSC gene editing technology in biomedical research. I'm Lily Fan, the manager in cell line and gene editing platform. I'm very glad to introduce the iPSC gene editing today. I will introduce the topic from the following two parts. Let's start with some basics. What is the iPSC technology? Induced pluripotent stem cells are artificially induced stem cells. The induction process involves introducing reprogramming factors into somatic cells to revert them to an undifferentiated state and induce pluripotency. This great groundbreaking work was done by Yamanaka, who was awarded the Nobel Prize in 2012. So regarding the somatic cell selection, skin fibroblasts and blood cells are most used because the techniques for obtaining are well developed and mature. There are various delivery methods for reprogramming, such as integrating virus, integrated non-virus, non-integrating virus, non-integrated non-virus, and now the Sunday virus and episomal vectors are commonly used. And what is gene editing? Gene editing is a precise technology that enables targeted modifications to the DNA sequence, allowing insertion, deletion, or replacement of genetic material at specific locations in the genome. The mechanism of gene editing is engineered nucleases recognize a designed sequence and cleave genomic DNA. The host cell activates the DNA damage response and induces either of the two DNA pathways in IHEJ or HDR pathways. The former pathway induces gene disruption and frame shift induction, whereas the latter pathway induces donor knock-in and substitution. Among the gene editing technologies, CRISPR-Cas9 offers the highest efficiency and affordability, with simpler construction and delivery compared to ZFN and TALEN, which require complex protein engineering. How to generate iPSC gene editing cell line. Here is the workflow of iPSC gene editing. We will first do gene analysis and strategy design, and then construct design vector, then transfect the vector to iPSC cells and analyze the cell pool efficiency. The transfected pool with high efficiency will be selected to single clone generation. It will take about 2 to 3 weeks for iPSC single clone generation. PCR and Sanger sequencing are used for positive clones screening. To validate the positive clones, we will do a second round of sequencing and confirm the clones. And what are the challenges in iPSC gene editing workflow? First is the low gene editing efficiency. Second is the difficulty to culture iPSC. Third is the low monoclonal formation rate. So, how to address these challenges? Several solutions are proposed. First, as the gene editing efficiency is low, a convincing strategy is required. We have AI system can help select the best strategy. You can input the gene and cell line into the system. Then the system will evaluate the target gene, including the gene lethality, copy number, and the background of expression. Our AI system can give the best strategy quickly after comprehensive evaluation. The low gene editing efficiency is partly due to the challenges of iPSC transfection. We have two electroporation instruments and optimized transfection protocols. The transfection efficiency and cell viability of iPSC were both greater than 50% in our labs. As we know, knock-in is more challenging than knockout. To address this, we developed Cyagen's proprietary alpha donor technology by optimizing the homology arms and delivery system. Alpha donor works well on various cell lines, achieving efficiencies of up to 87%. By using our alpha donor technology, knock-in success and homozygous knock-in delivery can be guaranteed. Regarding the difficulty of iPSC culturing, we have some tips for it. We have self-developed culture medium, strict culture conditions, and standard operating procedures, which are crucial for maintaining the pluripotency and self-renewal capacity of iPSCs for a long time. Here are some data for our iPSC, which are in good condition, and all QC tests for the iPSCs have been successfully passed. For the single clone generation, we have 4 methods: limited dilution, pig clone, FACS, and microfluidic. We suggest mild dissociation reagents to digest the iPSC into single cell and change the medium regularly. Okay, we have a complete system for iPSC gene editing. Here are some case studies for iPSC gene editing. The iPSC enhancer knockout cell line generated by Cyagen was utilized in a study published in Nature Communications. First, we designed pairs of guide molecules and transfected them into iPSC cells, successfully obtaining homozygous knockout clone with about 10 kilobase deletions, which were validated by PCR and Sanger sequencing. Additionally, pluripotency analysis was conducted on the knockout clones to confirm their suitability for downstream applications. Cyagen's alpha donor technology showed high efficiency in knock-in and point mutation in iPSC cells, achieving a homozygous Ki rate of 16% and a homozygous point mutation rate more than 30%, with fast delivery starting from 8 weeks. Regarding iPSC QC analysis, edited clones can be validated through Sanger sequencing, pluripotent analysis as default, karyotype analysis, and off-target evaluation are add-on service as requested. In summary, the key challenges in iPSC gene editing include low editing efficiency, difficulties in iPSC culture, and the low monoclonal formation rate. To address these issues, first, optimal editing strategy selected by Cyagen's AI optimization of transfection conditions and application of alpha-donor technology can improve editing efficiency. Second, maintaining iPSC pluripotency and self-renewal capacity requires the appropriate culture conditions and standardized operating procedures. The mild dissociation reagents to digest iPSC into single cells and optimize the culture conditions can improve the monoclonal formation rate. How to use iPSC gene editing to generate a disease model. Take central iris hypoplasia as an example. Researchers used iPSC as a disease model and found altered chromatin topologies caused by balanced chromosome translocation led to central iris hypoplasia. Here is the research framework. Through a genome-wide linkage scan, WES and/or WGS, nanopore, fish, karyotype analysis, and single sequencing, a balanced translocation was identified. To further explore the molecular mechanism, iPSC, human eyes, mouse eyes, and zebrafish was selected as the disease models. By using patient and wild-type iPSC, research found the rewiring of the 3D genome by the translocation, resulting in upregulation of APCD1. By enhanced knockout in iPSC from the patient, they found that the overexpression of APCD1 was partially rescued. Studies also assessed the potential roles of APCD1 in the iris of human and mouse as well as observing zebrafish. Here is the data using iPSC as the model. Clusters of enhancers were detected within two chromatin topologically associating abdomens on chromosome 6 and 18. In the patient sample, the translocation resulted in ectopic integrations between the enhancer cluster with the APCD1 promoter. APCD1 was the only gene with a statistically significant expression change by RNA-seq, which was confirmed by RT-PCR and Western analysis. And 10.6 kilobase enhancer region was knocked out in iPSC from the patient, which was generated by Cyagen. The knockout clones were validated by PCR and single sequencing. RT-qPCR results show that overexpression of APCD1 RNA was partially rescued in iPSC, with the enhancer knockout comparing with that in iPSC from the patient. So what we can provide for iPSC technology? Cyagen offers one-stop iPSC services, including iPSC reprogramming, gene editing, and differentiation with faster delivery starting from 8 weeks. Cyagen ensures high-quality services through self-developed iPSC culture medium and standardized operations. Gene editing efficiency can reach up to 98% with the comprehensive quality control. We have a professional scientific and project management team dedicated to supporting customers. Moreover, Cyagen has integrated technologies and products for cell biology research. Except iPSC services, we can also provide gene editing cell models, customer stable cell models by using lentivirus and PB vectors, virus package, functional assays, and ready-to-use catalog cell models. We have successfully delivered over 2,000 projects. The alpha-donor system enables high-efficiency knock-in applications. The advanced iPSC technology provides a one-stop solution. The universal stable cell engineering technology can meet any customer demand for drug discovery and development. Additionally, Cyagen provides the best value service for our customers. That's all for today's webinar. Thank you for your attention. We hope this session has provided you with valuable insights into the application of iPSC gene editing technologies in biomedical research. If you have any questions, feel free to contact us.
Okay, Lily, thank you for walking us through the iPSC journey from its genesis to its wide application, as well as what Cyagen can offer. I believe most customers know Cyagen as a company specialized in animal model generation, but your presentation really show our expertise not only limited to generating complicated mouse models, but also extend beyond that in iPSC. So I have some questions from the audience. Let's see what the questions are. Okay, the first one being, what are the challenges for iPSC gene editing?
Okay, the challenges for iPSC gene editing include low gene editing efficiency, difficulty to culture iPSC, the low monoclonal formation. So our solutions are first, optimal editing strategies selected by Cyagen's AI system, optimization of transfection conditions, and application of alpha donor technology can improve gene editing efficiency. And second, maintaining iPSC pluripotency and self-renewal capacity requires the appropriate culture conditions and standardized operating procedures. And third, the mild dissociation reagents to digest iPSC to single cells and optimize the culture conditions can significantly improve the monoclonal formation rate.
Okay, the next question is, what's the QC method used for iPSC gene editing?
Okay, so we use the PCR, sample sequencing, and pluripotent analysis for the positive clones as default. If client required, we can also provide a karyotype analysis and off-target analysis.
Well, it looks like we have applied very stringent QC. So the last question is, what's the turnaround time for iPSC gene editing?
Okay, the timeline is from 8 weeks for iPSC knockout and point mutation service. It is from 12 weeks for iPSC knock-in service. Okay, that's all.
Okay, so the timeline really depends on the nature of the mutation, and that makes sense. Okay, thank you again, Lily, and this concludes our webinar today. And thank you everybody for watching. We'll see you in the next episode. Bye-bye.
Okay.